Hello everyone, welcome to 10-Minute Physiology. In today's video, I'm going to try to explain to you how action potentials propagate down an axon in less than 10 minutes. So let's give it a go. So I would like to start off with a magnification of the axon. And like many other parts of the cell, the axon is characterized by a lipid bilayer or plasma membrane that separates two different fluid compartments. The first fluid compartment is the intracellular fluid, and the intracellular fluid, or the ICF, is the fluid inside the cell. The second fluid compartment is the extracellular fluid, and the extracellular fluid, by definition, is the fluid compartment outside of the cell. Now, you may remember from your university classes or from maybe one of my earlier videos that the axon has many different types of ion channels. However, in this video, we're only going to be focusing on one type of ion channel, and this will be the voltage-gated sodium channel. Now, the voltage-gated sodium channel has two different types of gates, the M gates and the H gate. So in the voltage-gated sodium channel, there are 3 M gates and 1 H gate. Now, the M gates are otherwise known as the activation gates, and activation gates are gates that open in response to depolarization, and they do so really quickly. The H gates are inactivation gates, and inactivation gates close in response to depolarization, and they do so slowly. If you need help with these gates, I have a video in my channel titled The Hodgkin-Huxley Model, Voltage-Gated Sodium Channels and Voltage-Gated Potassium Channels. I have a video for each of them. So if you have any trouble with these gates or the terminology I'm using, please refer to those videos. Now, at rest, what we will tend to see is that the M gates of the voltage-gated sodium channel will be closed and the H gates will be open. What does this mean? This means that the resting potential, voltage-gated sodium channels are closed but not inactivated. Now let's say that a graded potential starts moving down the axon. And when the graded potential moves down the axon, let's say it hits this voltage-gated sodium channel. So when this graded potential hits the voltage-gated sodium channel, it will cause the membrane in that region to depolarize or become more positively charged. So what is the result? The result is that this depolarization of this voltage-gated sodium channel will cause the M gates to open really quickly. Now, when the M gates open and the H gate is open, what's going to happen is that sodium will flow down its electrochemical gradient from the extracellular fluid to the intracellular fluid. And as a result, the cell will further depolarize. Now, when the cell depolarizes, what happens is that the H gates will eventually close. And this is because the H gates close in response to depolarization, but they do so slowly. Now, what's going to happen? Now this positive charge that has entered into the cell through the voltage-gated sodium channel will start moving down the axon, and it will go to the next voltage-gated sodium channel in line. Now, what's going to happen in this case? This positive charge will depolarize the voltage-gated sodium channel. This will cause the M gates to open, sodium to flow down its electrochemical gradient, and when sodium flows down its electrical chemical gradient, it will depolarize the cell. Now, in response to this depolarization, the H gates will close and the voltage-gated sodium channel is now inactivated. So now we have 2 voltage-gated sodium channels inactivated. Now, the positive charge that entered in through this voltage-gated sodium channel will move to the next voltage-gated sodium channel in line, open the M gate, and cause sodium to flow in, and the cycle keeps on going. So, in order to understand how action potentials propagate, we have to have a clear understanding of what's going on in this last couple slides. So here, what I have is a positive charge. And this positive charge has entered in through a voltage-gated sodium channel. Now, when the positive charge enters in through the voltage-gated sodium channel, what's going to happen is that this positive charge will move in 2 directions. The first direction will be in the retrograde direction. So in other words, it will be moving backwards. However, it's important to realize that the voltage-gated sodium channels that have already fired the action potential will be inactivated. So as a result, the positive charge that is moving from this voltage-gated sodium channel backwards to this voltage-gated sodium channel will not have any effect on this voltage-gated sodium channel. And this is because the H gates are closed. Remember that when the H gates are closed, the voltage-gated sodium channel is inactivated. And the only way to reactivate it or otherwise open that H gate is a hyperpolarization. So by doing this, we prevent an action potential from firing in the retrograde direction. So in other words, we prevent action potentials from moving backwards. The other direction that positive charges will flow is in the anterograde direction, or otherwise known as the forward direction. So what in this case, the positive charge will move from this voltage-gated sodium channel to this voltage-gated sodium channel, and it will cause the M gates to open in this case. And when the M gates open, it will cause the action potential to propagate and fire from this region. So in other words, what we see here is that the action potential will only propagate in one direction. And what keeps it from propagating in the backwards directions is the inactivation of voltage-gated sodium channels behind it. So the inactivation of these voltage-gated sodium channels prevents the backwards motion of action potentials. So what this mechanism does is it keeps action potentials moving down the axon in one direction towards the synaptic terminal, which will then be used to release neurotransmitters and cause synaptic transmission, which we will talk about in a future video. Now, an important concept to understand is that a lot of our neurons in our body are myelinated. And myelin is basically can be seen as an insulator. This insulator basically prevents charge from leaking away from the intracellular fluid. So in other words, it helps voltage to spread further down the axon. Now, when we're talking about the axon and myelinated axons, there are specific regions called the nodes of Ranvier. And the regions that are unmyelinated are nodes of Ranvier. So these little white rectangles that we see here are the nodes of Ranvier. And the nodes of Ranvier are where the voltage-gated sodium channels are. So when we're depolarizing the cell, the voltage-gated sodium channels will open in the nodes of Ranvier, and it will allow sodium to flow into the cell. And then the sodium will travel down the axon to the next node of Ranvier and cause an action potential to fire there. So in other words, what we see is that the action potentials will fire in these nodes of Ranvier. And these myelinated sections are just meant to keep the charge from leaking out of the cell. So it allows the charge to spread further, and it allows there to be a stronger depolarization in the next node of Ranvier. So in other words, what we see is that the signal will sort of hop from node of Ranvier to node of Ranvier. So the signal, let's just say we have an action potential in this node. This would cause an action potential. The charge would spread all the way from this node to this node and cause an action potential to fire here. The charge would spread and cause an action potential to fire here. So in other words, the action potentials will hop from one node to the other. And this is called saltatory conduction. In other words, the action potentials are hopping from one node to another. And this is because the voltage-gated sodium channels are only present in the nodes. So I hope this helps you understand the propagation of action potentials a little bit better. If you have any questions, please ask them in the comments section. I hope to see you in the next video, and good luck in your studies.